Thank you for the introduction and thank you for the invitation. It has been a really fruitful day to discuss with all of you. Thank you very much. I hope I still have energy to give my talk now. So today it's a kind of hybrid talk with two parts, but the two parts are about anthropogenic hybridization in marine altered habitats. So one part will be on pontoon sea vase or tunicates, sayona. So these tubes here. And the other part will be about mussels, standard mussels that you can eat with fries, but not this one. They are in polluted ports. Okay, so usually people interested in hybridization works in hybrid zones. And as said Godfrey Hewitt, there are natural laboratories for evolutionary studies or windows to the evolutionary process. I've shown a few pictures of some of the most famous hybrid zones, but there are many others. Most of the time, these hybrid zones are post-glacial secondary contacts. So you have one lineage on one side, another lineage on the other side. They came into contact and they meet in a narrow hybrid zone. The contact is often a few thousand years, so post-glacial. And sometimes you can study different transects of the same hybrid zones. You can gain some information, but it's usually mainly the same big story because you have the same genomes each side of the contact zone with a similar demographic context, which is a balanced demographic context, two big populations of lineage 1 and lineage 2. But now, as you are all aware, people, humans, move the biodiversity, shake the biodiversity all over the world. And it is mainly due to trades and the maritime traffic. Here you have a map of the maritime traffic all over the world. You see all these connections. So you can have terrestrial species that are trade, and you have many, many invasive species that colonize new places worldwide. But for marine species, they can hitchhike directly in the ballast water or even on the ship hull. For example, mussels, they are part of the biofouling community, so they can travel by boats. The difference with post-glacial hybrid zone is the age of the hybrid zone. It is very recent. And rather than calling them anthropogenic, we can call them anthropocenic, the hybrid zones of the Anthropocene. They are very young, a few decades, most of the time after the World War II, when all the maritime traffic exploded. And you see that the contacts are very different because you take an alien lineage, sometimes from several places of the source distribution at different places of the inside the native distribution with different ecological context and different demographic context. So to me, they are really my new playground to study hybridization. So why they are interested to study hybrid evolution? It is because you mix together two parental genomes and you obtain, after the first generation of hybridization, you obtain a mosaic of chromosomal blocks inherited from one lineage or the other lineage. And you can study it like an evolutionary experiment, not as controlled as what you do here with flies, but nonetheless, it is at a scale that you cannot have in the lab. Imagine a port. It's a super big pool. No grants can pay such a pool to make evolutionary experiments. So I use this as an evolutionary experiment. It is not so much controlled, but it is the scale is quite bigger. So what can we do with these mosaic genomes? We can describe the mosaic of ancestry. We can look at the individual ancestry at individual level, which is the proportion of parent 1 ancestry and parent 2 ancestry in an individual. Or you can look at the local ancestry and more precisely, the average local ancestry of the population sample. So for a chromosome position, you look at the average local ancestry along the chromosome. And what you expect is to find regions of the genome that are fixed for one or the other parental ancestry. If you have a desert of ancestry, so a region that is fixed for the major parent, the parent that contributes the most to the unmixed lineage, you expect this region to be the place of a genetic incompatibility. And usually it works very well. You have 3 examples here. Unmixed population in swordtail fishes, in sparrows, and in heliconius butterflies. And you can see all these deserts of ancestry where you expect to have genetic incompatibilities, and actually often they coincide with genes that are known to be involved in reproductive isolation. You can also have only a small part of the genome that is enriched for the alien ancestry, making a peak of the minor parent ancestry or a peak of the alien ancestry. And this is expected to coincide with adaptive introgression, an alien allele that is adapted in the recipient genetic background and the local environment. And again, I show you a nice example in Fondulus killifish. So in the Houston port in Texas, you have heavily polluted populations full of hydrocarbons. And here you have a bay or lagoon that is non-polluted, quite clear. And a resistant allele, which not resistant, tolerant allele. This allele makes the fish tolerant to hydrocarbon pollution. It is due to a deletion that knock out hydrocarbon receptor. These alleles come from another species, Fondulus heteroclitus, and has introgressed into the genome of Fondulus grandis. And here you have the allele frequency, well, the genotype frequency climbs between the polluted populations and the non-polluted populations. So the introgressed allele is favored in the polluted sites and must be costly otherwise and make this climb. What I want to emphasize here is that most of the theoretical models that describe adaptive introgression consider a single recipient population, what I call global introgression. And most of the experimental examples of adaptive introgression found local adaptive introgression, where the introgressed allele is adapted only in polluted populations or in where you have insecticides or things like that. And it is only the advantage of the foreign allele is just local. And it makes an important for the genomic footprint of selection. Okay, so now I come to my two examples, my two tales of anthropogenic hybridization that are actually the two tails of the possible outcome of anthropogenic hybridization. One is in Sayona intestinalis as a native species, Sayona robusta as an invasive species. Introgression is found in marinas, leisure ports, and it is an example of adaptive introgression. And the other example is an example of a new ecotype of mussel that lives only in ports. The native species is Mytilus edulis here, and the invasive species is Mytilus galloprincialis, and admixture populations are found in big commercial ports. And it is an example of whole genome admixture. And before to start, I would like to say that in the two cases, we are at the end of the speciation continuum. The two parental lineages are very divergent, both in molecular divergence and in molecular times. Here you have the result of a study that evaluated the probability of gene flow between populations as a function of molecular divergence, the absolute divergence, so the fixation between populations. When divergence is low, it's one species. You can have a gene flow all over the genome. And I put Neanderthal and Sapiens as an example. At the end of the continuum, you have true species without any gene flow in the genome. And in the middle, you have the gray zone where you have semi-isolated species with some region of the genome that can be exchanged and some region of the genome that are blocked by barrier loci. And the muscle are here, 2% nucleotide divergence, 2 million years. And the Sayona quarks, the divergence, 15%, is the same as between this monkey and the ant that carry it, which are humans. But they look very similar morphologically. Okay, so I have three parts. First, adaptive introgression in pontoon vase tunicates. Second part, full admixture in the duck muscles. And finally, I will show you a multigenic model of hybrid fitness that can explain the results we obtain in muscles. First, tunicates. So the alien species is Sayona robusta. It comes from Asia. It has been introduced in Europe in the range of Sayona intestinalis. They hardly hybridize in the wild. They hybridize in the lab quite easily, but in the wild, very few hybrids. But we found regions of chromosome 5 with a peak of local ancestry of the robusta parent. This is the fact that it is adaptive introgression has been validated by several tests of selection, volcanofinder, extended homozygosity test. We dated the age of the introgression to 75 years, which is roughly what we know about the introduction of the Asian species into Europe. And by increasing the sample size, we narrowed the center of the peak to only 3 genes. And among these 3 genes, we have cytochrome P450, a CYP gene that can be a candidate for cellular detoxification. So we used long-read sequencing to try to better understand the structure of this polymorphism. And we found that the Robusta allele is actually a tandem repeat of 23 copies of the gene, while the intestinalis allele has just one copy. So if you want to cross the 23 repeats, it's very difficult, and we did not manage actually to cross it. The biggest read we obtained of this region was 138 kilobase, and we just obtained 20.5 copies. So we don't know exactly the number of copies. What we know is that each copy has exactly the same nucleotide sequence, and each breakpoint between copies is exactly the same. So you cannot use an assemblage to scaffold the number of copies. It's not possible. It's exactly the same copies, the same copy 23 times. So we use the coverage as an estimation. If you use short reads, you have a correlation, but you do not estimate very well the number of copies. But with long read, we found that the homozygote for the intestinalis allele has 2 copies, one in each parental chromosome. The heterozygote have 24 copies, one in the intestinalis chromosome and 23 in the Robusta chromosome. And the homozygotes for the Robusta allele have 46 copies, 23 copies in each parental chromosome. We have a little variation of +1 or -1 copy. We don't know if it is the estimation or if it is true variation in copy number, but you can ask me the question at the end. I will give you my opinion. Okay. So next, we wanted to have a nice estimation of the local ancestry blocks on the chromosomes. It's quite expensive to use long reads on many individuals. So we use a sequencing protocol, which is haplotagging. The idea is that you use long DNA molecule, you put them into beads, and you add tags, molecular tags that give you the information of where the short reads come from. So it's a method of linked read sequencing. And to infer local ancestry, we use this method, LOTER, but other methods give very similar results. And at the end, you have the result. So in blue, you have the intestinalis ancestry, and in purple, the Robusta ancestry. So you see here the location of the adaptive introgression and various lengths of introgression tracts. And here you have the frequency of the introgress alleles in the different populations. And you can see that first, we don't have fixation. And second, we have difference in allele frequency between populations. So why do we have this mosaic structure? To better understand it, we contrasted two models. One is a transient selective sweep model, where you have introgression from species A into species B, and a stepping stone population structure in species B. So the adaptive allele sweep from population 1, population 2, population 3, population 4. So it reached a higher frequency in population 1 first, and while it propagates, it erodes the Robusta ancestry because of recombination with the local intestinalis chromosome. And so at the same time it propagates, the introgression tracks become shorter. And it creates a correlation between the frequency of the adaptive allele, which inform about the age of the migration of the adaptive allele, and the length of the introgression tract. And it gives a positive correlation. Actually, it always gives a positive correlation, because if migration is too high, you have the same frequency in every population. The frequency increase similarly in all the populations. If migration is too low, you have fixation in some populations and no polymorphism in other populations. If you want polymorphism in every population, you need a balance between migration and selection that creates the difference in allele frequencies between population. And in this case, you always have a correlation. In the second model, you have introgression of an allele that is adaptive only in the blue environment, in the blue habitat. And it is deleterious in the red habitat. We explored various population structure. I won't explain it now. But what happened is that what affects the allele frequency is local selection, high frequency in the positive selection beams and low frequency in the negative selection beams. It's not the timing, it's local selection. And so you have no correlation between the frequency of the adaptive allele and the length of the introgression tract. And what we observe in the true data is no correlation between the frequency of the adaptive allele and the length of the introgression tract in accordance with the local sweep model. Yeah. At the end, it is sunny at the end of the day. Okay. Okay. So I continue. I am finished with tunicates, Iona now, and I am going to explain you what we observe with muscle. So to talk about muscle, I have to explain that I have two species in Europe. Sorry. Mytilus edulis, where is it? Here, in blue, and Mytilus galloprincialis in red and yellow. But in Mytilus galloprincialis, I have a Mediterranean lineage and an Atlantic lineage. And they meet in a hybrid zone here in Algeria with very few hybridization. So even though at the taxonomic level it's the same species, at the genetic level it's like two semi-permeable species. And what we found is that in some commercial ports of France, Saint-Nazaire, Brest, Saint-Malo, Cherbourg, Le Havre, ports that look like this, you know, very big ports. We have admixed mussels. Sorry, I have problem with the pointer. Yeah, it's here. Between not the Atlantic lineage of Gallo, but the Mediterranean lineage of Gallo. So here you have Mytilus edulis, Gallomed, Gallo-Atl. Here you have natural hybrid zones between edulis and Gallo-Atl. And here you have the dock mussels, the admixed lineage found in commercial ports. And after that, we analyzed new ports in Dunkerque, Rotterdam, Swansea, and we found more introgressed dock mussels with a higher proportion of edulis ancestry. We also found that they shared a common genetic composition So it's not several independent admixture, it's a single admixture and then propagation from ports to ports thanks to the maritime traffic. And if we look at the local ancestry along the genome in the southern port or in the northern port, we have what I think is a flat landscape of ancestry. At least we have no fixation anywhere in the genome. We have no desert and most importantly, we have no desert of major parent ancestry. So I must recall here that the two parental lineages are very, very divergent. So to me, it's a big surprise. We also found no correlation with recombination rate, no correlation with chromosome size. And here you have the difference of ancestry between the northern port and the southern port. You expect to have a resistance to introgression in regions with genetic incompatibility. And again, you have no resistance to introgression. It's as if the full ancestry moves up to 20% more edulis ancestry in the north. So very surprising. And finally, we can look at the individual ancestry. So here, each point is an individual muscle. And on the x-axis, you have the ancestry proportion. And on the y-axis, you have the ancestry heterozygosity. And you see that you have this line of genotypes going from the Gallo parent to the Edulis parent, with more Edulis ancestry in the northern port in blue. So how can we explain this? We cannot explain this with simple Dobzhansky-Muller incompatibilities. We cannot explain this with local adaptation like port alleles. It does not work. They should fix as well. So we need something more complex with more epistasy, more redundancy, more context dependence. So we use the Fisher's geometric model. So to recall what is the Fisher geometric model, it is a quantitative genetics model with many traits. So not just one trait, but many traits. Here you have an example with two traits, trait 1 and trait 2, but in reality you have in the model n quantitative traits. You have optimizing selection. It means that you have an optimum here in the middle and a gray shading going from the fittest place, the fittest area of the multivariate trait to the lowest fitness. And mutations are vectors, arrows in this landscape, in this multivariate space, right? So for example, this mutation is a positive mutation as it increases both trait 1 and trait 2 value. And from this genetic background, it makes the genotype closer to the optimum. But this exact same mutation in another genetic background can be a neutral mutation. And the exact same mutation in another background can be deleterious. So this is something we want in speciation research. It is the spirit of the Dobzhansky-Muller model. When one mutation can be positively selected or neutral in a given genetic background, but deleterious when you put it in another genetic background. You can also move the environment, so the optimum, to make this deleterious mutation a positive mutation. So you have context dependence. So two properties that are important for us. Then how you make divergence? To make divergence, you start from an ancestor somewhere and you accumulate mutations. So in parent 1, each time you have a new mutation, it makes a derived allele in parent 1 and you keep the ancestral allele in parent 2. So we have 4 mutations in this example, from the ancestor to parent 1. We make the same for parent 2, 3 mutations, and each time you define a new locus in the genome. So here it is the genome, a single chromosome. We need diploid genomes because we are studying diploid species. So you just assume additivity of the mutation at the phenotypic level. And then to make a hybrid, you randomly sample mutations from parent 1 and parent 2. If you exactly sample one mutation of each parent to make a F1 hybrid, you have the mid parent, so the F1 is exactly in the middle of parent 1 and parent 2, and it is fully heterozygote in the genome. If you make recombinant hybrids, then you can go in wild fitness phase, especially if you use homozygote mutations. You have a low fitness of hybrid. So we obtain what we want when you study two diverse species. But you can also make hybrid genotypes that are fitter than the parents, which is also something that we want. And you can make them with different combination of mutations, which means you have genetic redundancy, which is also something that we want. So the Fisher geometric model has all the characteristics we need to study hybridization, to have hypostasis, genetic redundancy, and also to change the optimum of the fitness landscape. So what we predict is that if, so it is in this paper, Simon et al. 2018, if the two parents are well adapted here, you expect selection for the major parent ancestry, and you also expect heterosis. So only the F1 or genotypes that are here have a high fitness, similar to the parent, but you cannot maintain heterozygosity in formictic populations. So it is a non-stable equilibrium, and so you either select for P1 or P2. You can have a local selection if one parent is adapted and the other parent is maladapted. You mostly select for the fittest parent, but you still have bistability, which is if the less fit parent start with a higher proportion than an equilibrium proportion value, you can still select for the unfit parent. Bistability is what you expect in hybrid. But now, if you change the environment, you move the optimum, then the two parents are maladapted. And in this case, selection for parental genome is much less efficient. And so you can select, in this case, for unmixed genome. So briefly, I added some simulations. So in this case, you start in this is the 5th generation. You have individuals in this space, and quickly you select for P1 in this case. Okay. Under the exact same parameters condition, you now change the optimum, and this time you see the yellow color, which means individuals are maladapted to the new environment. And instead of selecting for P2, sorry, you stay with unmixed proportion. So we obtain, I think, what we want. that if you have an admixed population in a new environment where the two parents are not adapted, you maintain admixture in the genome. So I come back to this triangle with the true data, and you can say if I sample the muscles, it means that they were fit. So just try to fit the model, and this is what we obtain. Here you have the iso fitness line of the fittest parent, and the dock muscles are a little bit above this iso fitness line. It is in accordance with the idea that parents are maladapted to the port environment, that although you have selection for the fittest parent, which is Galloprantialis in this case, selection still maintain admixed genomes because we have heterosis actually in the genome. But it's not perfect because I don't have phenotype. And to finish with, just a try with a phenotype, I use the microbiome. The idea is that the microbiome diversity can be used as a fitness proxy. If a muscle is healthy, it maintains a diverse microbiome. If it is not fit, opportunistic bacteria can invade the microbiome. So here you have the distribution of the microbiome diversity with the Shannon index. Here you have 1 minus the departure from the mean microbiome diversity together with the PCA that shows that most of the muscle have a similar microbiome with a high diversity, and some muscle have deviant microbiomes with low microbiome diversity. So these muscles are expected to be fit, and these muscles are expected to be unfit. So you can plot the phenotype in this triangle and then fit the model, and you obtain a better estimation of the fitness surface. And again, you see that we are in a fitness surface where you expect selection for heterozygous genotypes. So here are my conclusions. So ports are Darwinian playgrounds to study hybridization in the Anthropocene. Pontoon sea vases adapt locally in the port thanks to a ship tandem repeat introgress from an alien species. The dock mussel is a new port ecotype with a fully admixed genome. That I mean a lack of ancestry fixation, which is a big surprise for two species that are so divergent. So to explain this result, a polygenic model of stabilizing selection involving epistasis, genetic redundancy, and context dependence may explain genome-wide ancestry polymorphism in an admixed population in a new environment. And I would like to thank my collaborators, Manon Le Goff, she's a PhD student, and Frédéric Cerquiara, our engineer. You see how ports are playgrounds for us. Alexis Simon, who did the theoretical work together with John Welsh, my long-term collaborator at the University of Cambridge. Fanny, who genotypes the muscles, and Frédéric Viard, who introduced the idea to study ports and adaptation to ports in our team, and the INR for funding, and you for your attention. Thank you very much.